Hello and welcome to my channel. So in today's video, we are going to discuss about microbial growth and its control. The bacterial cells require certain nutrients, macronutrients as well as micronutrients for its growth. Macronutrients are those nutrients, which are needed in large amounts like carbohydrates. And micronutrients are those which are needed in small amounts. A growth media consists of carbon source, energy source, nitrogen source, inorganic salts, and trace elements. A defined media is a media which has known exact amount of ingredients, whereas complex media composition is not known. Although it is made up of yeast extract, beef extract, and peptone only, but we do not know the exact composition. Let's understand how inoculation of culture is done. This is how an inoculation loop looks like. This is your bacterial plate. With the help of this loop, we can just pick up a culture and put it in the liquid broth. So inoculation is the process of adding bacteria to a culture media so that they can reproduce over there. Inoculating loops introduce microorganisms like bacteria, yeast into plated or tubed growth media before incubation and multiplication or growth. This is a spreader. Spreader helps in spreading the bacteria evenly over the Petri plate, which has growth media in it. So spreader is an instrument with a long handle and smooth head designed for spreading and dispersing liquids evenly across the surface of agar culture plates. They can be made of glass, plastic, or metal. Right conditions like food, correct temperature, microbes can grow very quickly. Now let's learn about bacterial division. Bacteria and archaea, they reproduce asexually. So only while eukaryotic microbes can engage in either sexual or asexual reproduction. Now, bacteria and archaea mostly commonly engage in a process known as binary fission, that is, mother cell divides into two, where a single cell splits into two equally sized daughter cells. So this is the growth curve pattern of a bacteria. This graph actually tells us about log of number of bacteria versus time. There are 4 phases: lag phase, exponential phase, which is also called log phase, and stationary phase, followed by death phase. As we can understand from the graph, the lag phase is a time when bacteria is inoculated, in the new environment. It is still adapting to its surroundings. Exponential phase, log phase, is the phase where bacteria is dividing. Stationary phase is the phase where no more new bacterial growth is there and bacteria has also started dying, but the number of growth is equal to number of death. And death phase is when the nutrient, the media has started secreting toxins, and now the growth of bacteria has declined. Lag phase is an adaptation period where the bacteria are adjusting to their new conditions. The length of the lag phase can vary considerably. Typically, bacteria may take some time in adjusting to the new environment. Exponential or log phase is the one where doubling of the population takes place in an exponential manner. And in this manner, cells are the healthiest and most uniform, which explains why most experiments utilize cells from this phase. Generation time is used to calculate the time for the bacterial population to double in number. So each bacteria has a different generation time. Stationary phase means the number of cells being produced is equal to the number of cells dying off, resulting in a flattening out of growth on the growth curve. Death phase or decline phase is the number of viable live cells decreases in an exponential fashion. It has been shown that 100% cell death is unlikely for any cell population as the cells mutate to adapt to their environmental conditions. However, often there is tailing effect observed. Disinfection means the process of eliminating or reducing harmful microorganisms from inanimate objects and surfaces. This is called disinfection. And sterilization is a process that removes, kills, or deactivates all forms of life. And the sterile products that undergo sterilization are often chemical, heat, or filter sterilized. That's all for the video. Thank you.